All right, so now we're going to start the process of cellular respiration. And just a reminder, our objective here is to create a model of respiration and be able to tell the energy that comes in and out. We also have an additional objective today, which is we are going to model anaerobic respiration and explain why more glucose is used and why it happens. So we're starting our first objective, which is to model aerobic respiration. But in starting that, we're actually going to be able to compare anaerobic with aerobic. So first, we need to recognize where we are in the cell. We are in the cytoplasm of the cell. Now, just FYI, I am actually going to use the same doodle notes for the whole process. So if I were you, I would get a piece of paper, turn it the wide way, horizontally, and I would try and get all of this, the next 3 videos, all on one page so that you can see how it's all one process. All right, so we're going to start off in the cytoplasm. And we have our sugar, C6H12O6, our glucose. And I just drew this up here so that you could see that we have 6 carbons. And we're going to follow these carbons very closely as we move on. This is going to undergo the process of glycolysis. Remember, glycolysis is literally breaking the sugar. Glyco meaning glucose, lysis meaning to break. So I just broke this 6 carbon sugar into 2 3 carbon compounds, which are called pyruvate. Now, how did I do this? I did this by, remember how the saying, it takes money to make money? Well, it takes ATP to make ATP. So we're going to actually put into the process 2 ATP, and in the process we'll end up making 4 ATP once we break up these compounds. We are also going to make something called NADH, which I don't necessarily want you to know, but we need to call them something because they're going to be very important later. So I am going to call them high energy compounds, and I will abbreviate that HEC. So basically we are going to make these high energy compounds as well. And I didn't leave a lot of room. Plus high energy compounds, HEC, high energy compounds. All right, now these are going to be very important later because they're going to contribute a lot of hydrogen that we need. Okay, so that's glycolysis. So based on what I just told you, I wrote out a chemical formula. C6H12O6, or glucose, plus 2 ATP makes 2 pyruvate and 4 ATP. Now, you guys haven't had chemistry yet, but in chemistry, if you have things that are on the same on both sides, you try to reduce them. So in this case, we have 2 ATP on this side and we have 4 over here. So really we netted, or we ended up with in the end a net addition of 2 ATP. So glycolysis creates 2 ATP is made. And this will be important later when we tally up how much ATP we made. All right, so now we're going to take a little diversion. So at this point, we haven't used any oxygen. So glycolysis is technically part of anaerobic respiration. So I'm going to take a pause and I'm going to talk about anaerobic respiration. If I were you, I would leave all of this right here and flip your paper over to do the anaerobic respiration portion. What is anaerobic respiration? Very simply, it's AN. This means without aero oxygen. So this is respiration without oxygen. So like I said, so far we haven't used any oxygen and we are still in the cytoplasm. So at this point, depending on what organism we're in, is going to depend on what type of anaerobic respiration we undergo. So at the end of glycolysis, we have 3 pyruvates, and we have 2 options of where we want to go from here. The first option is to take that pyruvate and turn it into lactic acid. When you do that, we use lactic acid in our muscles. When we are not getting enough oxygen when we are working out, there's not enough oxygen to continue on with cellular respiration normally. So our muscles undergo lactic acid fermentation. That's why I drew a little muscle here. When that happens, this conversion from pyruvate to lactic acid, some of these high energy compounds are released out, and that gives the process the energy it needs to break up more sugar into pyruvate. Also in that process, we're going to make 2 ATP. Oops. Okay, so overall between glycolysis and lactic acid, we're making 4 ATP, and we are getting those energy compounds that we need to break up that 6-carbon sugar into pyruvate and to continue this circle of creating lactic acid. Now, in your muscles, lactic acid, after oxygen is added, so after we get to catching our breath and we get enough oxygen, the lactic acid is broken down in the liver. And so usually it takes about 24 hours to 48 hours, depending on how hard the workout was, for your liver to process all of that lactic acid and for your muscles not to be covered in lactic acid. All right, so that is in an animal or more of a human system. Let's talk about bacteria. So our other option is we can take that pyruvate and turn it into ethanol or alcohol. When I take that pyruvate and I convert it into alcohol or ethanol, it's called alcoholic fermentation. And that also releases, just like we said before, it's going to release those high energy compounds so that we can go back and break up more of that glucose. And it also creates the same amount of ATP that lactic acid fermentation does. So in alcoholic fermentation, this is what we use to make wine and beer, because this happens a lot in unicellular organisms or bacteria and other things like that. So the glycolysis is the anaerobic part of cellular respiration. But if we continue to not have oxygen available, then the choices are lactic acid fermentation or alcoholic fermentation. And the reason why more glucose is used, because look how quick this cycle is. It goes glycolysis, lactic acid, then break up more sugar. Glycolysis, lactic acid, break up more sugar. It's very, very fast. So a lot of sugar is getting broken, but it's very inefficient because we're not making very much ATP. We're only making 2 from the lactic acid, right? So it is not an efficient process. It's not going to give us very much energy compared with the 38 we'll get at the end of cellular respiration. However, more sugar is broken up quicker in the lactic acid and the alcoholic fermentation.